Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
For decades, the way we thought about muscles working, down at the molecular level, that little motor, myosin, with its swinging lever arm, it was basically just, a cartoon in textbooks.
Right, really important cartoon.
Yeah.
We knew it had to swing to generate force, but actually seeing that specific moment, capturing it in atomic detail, that was the missing piece.
Until now, because some incredible new research has finally filmed it, they actually caught that fleeting movement, proving what scientists have theorized for ages.
Pretty amazing stuff.
Welcome to the deep dive. Today, we're jumping into a really cool breakthrough in molecular biology. We're looking at a study from Nature that used this cutting-edge technique called time-resolved cryo-electron microscopy. Basically, they made a movie of myosin in action, and our mission here is to unpack this pretty complex science, pull out the key insights, and help you understand not just what they discovered, but really why it's such a big deal, turning these tiny molecular details into something tangible.
Absolutely. And it's wild how long this specific step remained. Well, unseen. Myosin motors, these tiny powerhouses, they drive almost everything, right? From you lifting a weight
Big muscle contractions.
to really intricate movements inside your cells. They're like little engines converting chemical fuel ATP into a physical pull. A swing of this lever arm pulls on actin filaments, the cell's ropes, making things move.
And like you said, we had pictures. Scientists got these static snapshots, mostly using X-ray methods. You could see myosin before, all primed and ready.
The pre-power stroke state.
Right. And then the after state, once it had done its work, the post-stroke. But that critical moment in between, when it's just grabbed onto actin, still primed, just about to swing, that was the ghost in the machine.
Totally elusive. It was a huge gap in understanding how force actually gets generated. You missed the crucial transition.
So the big problem was speed, right? How do you film something that happens in milliseconds?
Exactly. Your standard structural biology tools, they're just not fast enough. It's like trying to photograph a hummingbird's wings with an old box camera. You just get a blur, if anything.
Okay, so how did these researchers crack it? Sounds like it took some serious creativity.
It really did. It was this beautiful combination of clever biology tricks and brand new tech. First, the biology side. They actually engineered a special version of Myosin 5.
Engineered it, like modified it.
Yeah, double mutant. Think of it as a slightly sticky motor.
Okay.
They designed it so it would hang on to actin just a little bit longer in that elusive primed state.
Ah, slowing it down intentionally.
Precisely. And crucially, it also slowed down one specific chemical step, the release of a little molecule, inorganic phosphate, or pi. That bought them precious extra time, milliseconds, which is like an age at this scale.
Okay, so they slowed down the action. Then what about the camera?
That was the tech leap. Time-resolved Cryo-EM. They use this custom-built microspray gadget. It sounds complicated, but basically it lets them mix the myosin and actin together and then instantly freeze them, vitrify them, turning them into glass without ice crystals just milliseconds later.
Wow, like freeze-framing the action.
Exactly. They specifically captured snapshots at 10 milliseconds and 120 milliseconds after mixing. Super fast, high-resolution pictures of molecules caught mid-movement.
And it worked. They could finally see that prime state clearly, separate from the post-stroke state.
They could. And what was really fascinating was they didn't just see two fixed poses. The data suggested a whole continuum of rotation for the lever arm, like seeing the entire arc of the swing, not just the start and end points.
So it directly proved that swinging lever idea we all learned. It wasn't just a diagram anymore.
It's real. They visualized it. That textbook cartoon came to life.
Okay, so let's dive into what they actually saw. In that very first moment, that primed state where myosin first touches actin, what does that look like?
Well, the first thing is, it's a pretty weak connection initially. Myosin mainly uses what we can call its lower half, the lower 50 kilodalton domain, to make contact. And importantly, the part of myosin that binds actin, this cleft, it's still open.
Like its jaws aren't clamped down yet.
Exactly. It's more of a tentative touch. Certain loops and structures on myosin make these first contacts. The upper 50 kelvin endodomain isn't really involved yet.
Interesting, just a light initial contact.
But, and this is really cool, they saw the very beginning of the actin protein, that's N-terminus, interacting with parts of that myosin lower domain. These seem to be the first little nudges that set everything else in motion. And in this state, the myosin lever arm is pointing sort of along the actin filament, angled, ready to go.
Okay, so actin makes contact. How does that initial, maybe weak binding actually prepare myosin for the big power stroke? What changes inside the myosin molecule itself?
Yeah, that's key.
Yeah.
Because it's not just myosin grabbing actin. Actin talks back. By comparing myosin before and just after it binds actin, they saw these subtle but really important shifts. That upper half of myosin, the U50 domain, actually rocks back slightly, sort of rotating around the actin.
Rocks back towards the main body of the myosin.
Exactly. It's not a huge movement initially, but it pulls on other internal parts, like rearranging things inside.
And that rocking has a purpose, right? It's not random.
Not at all. This rocking motion subtly rearranges key bits inside myosin's energy-binding pocket, where ATP binds and breaks down. It creates this internal strain, you could say.
A strain, like tension.
Yeah, like building tension. It positions the leftover energy molecule, ADP, perfectly for the eventual release of that phosphate, the pi.
Yeah.
Now, the exit door for pi, this backdoor pathway, it's still closed at this point. But that strain is building. It's like cocking a spring. So actin binding doesn't just provide an anchor. It actively primes the myosin motor internally, setting it up for efficient energy release. It's a real dialogue between the two proteins.
Wow. Okay, so the stage is set, the spring is cocked, then comes the main event, the power stroke. What did they see happen during that massive swing?
The biggest change, by far, is that lever arm just whips around. It swings a huge 93 degrees, mostly straight along the actin filament, very little twisting side to side.
93 degrees, that's massive on a molecular scale.
It is. And interestingly, that angle and the resulting movement, about 34 nanometers, perfectly matches how far myosin 5 is known to step along actin. So it's incredibly efficient, moving exactly the distance needed for its function.
And what about that cleft, the jaw you mentioned, the part that was open before?
It slams shut. As the lever swings, that upper domain rotates and the actin-binding cleft closes down tight. This creates a much bigger contact area with the actin filament nearly double the size.
Ah, so it goes from that weak touch to a really strong grip.
Precisely. It needs that solid anchor to actually pull and generate force without slipping off.
And this whole movement, the swing, the clamping down, that's tied to releasing the energy, right? Getting rid of that phosphate.
Absolutely. It's all beautifully coordinated. The rotation of that upper domain and the closing of the cleft physically push other internal parts around. This movement literally pries open that backdoor pathway we talked about.
So it squeezes the pi out.
It effectively squeezes the pi out of the active site. It's this mechanical action that drives the chemical release, completing that part of the energy cycle.
Okay, so putting it all together, the refined picture of force generation is, well, it's more complex than just lever swings.
Much more. Myosin binds weakly first. Then actin binding causes that upper domain to rock back, creating internal strain. Then, as the upper domain rotates fully and the cleft closes, that movement simultaneously opens the pi escape hatch and twists another internal part, called the transducer, which directly triggers the big lever swing.
It's like a Rube Goldberg machine, but elegant.
Ah, yeah, a very elegant one.
And this leads to that chameleon analogy they used, which I thought was brilliant. Can you explain that? It sounds really important for how myosin works under load.
It really is a key insight. Think about the directions of movement. Those initial actions, the rocking back of the upper domain, the closing of the cleft, they happen mostly sideways, almost perpendicular to the final power stroke direction along the actin.
Okay, so sideways clamping first.
Exactly. Myosin effectively clamps itself firmly onto the actin before it generates the main forward force. So when that lever does swing forward, especially if it's pulling against resistance, the force itself doesn't tend to pry the cleft back open.
Ah, like a chameleon gripping a branch tightly with its feet sideways before it moves its body forward.
That's the perfect analogy they used. It ensures a super secure grip before the power stroke really kicks in. This makes myosin incredibly robust, especially under load. It explains how different types of myosin can work so efficiently without slipping.
That makes so much sense. And you mentioned myosin 5-stepping. This efficient, secure movement must be crucial for how it walks along actin-carrying cargo.
Definitely. That large, mainly linear step of about 34 nanometers Fits perfectly with Myosin 5's job as a cellular delivery truck, and for myosins that have two heads or legs that walk.
Like tiny molecular legs.
Right. This precise, directed step means the detached head doesn't just flail around randomly trying to find the next binding site. It's guided quite precisely, making the whole walking process much more efficient and less likely to stumble, so to speak.
This really feels like a landmark study, taking that textbook diagram, the swinging lever, which has been around for, what, 50 years?
At least.
And finally providing the direct atomic level proof. Seeing it happen, that's a fundamental shift in understanding.
It is, and it's also a massive win for the technique itself, for time-resolved cryo-EM. It wasn't just a tech demo. They used it to solve a long-standing biological problem. It shows this technology is ready for prime time, ready to tackle other really fast transient biological processes.
So we can expect to see this used more now, to look at other molecular machines.
Oh, absolutely. This opens the door. Think about ribosomes building proteins or DNA helicases unwinding DNA. These are also incredibly fast dynamic machines. Researchers can likely adapt these strategies using mutants to slow things down, using these fast freezing techniques to capture snapshots of those processes too. The molecular biology toolkit just got a serious upgrade.
And beyond just understanding the fundamentals, you mentioned potential real-world applications.
Yeah, and this is where it gets really exciting for medicine. Being able to see these precise movements, how myosin works at an atomic level, and importantly, how mutations might change those movements. That's crucial.
Because some diseases are caused by faulty myosin.
Exactly. Certain heart conditions, cardiomyopathies, are linked to specific myosin variants. Some forms of deafness, too. If you understand exactly how a mutation breaks the machine, you can start thinking about designing drugs, precision therapeutics, to specifically fix or compensate for that defect.
So understanding the normal mechanism in this detail helps pinpoint what goes wrong in disease.
Precisely. It provides a structural blueprint for developing targeted therapies. It connects that deep fundamental science directly to potential clinical benefits down the road.
Amazing. So summing up, we've seen this incredible story unfold, clever biochemistry meets cutting-edge tech to finally visualize myosin's power stroke. It's not just a simple swing, but this intricate, coordinated molecular dance.
Yeah, every little shift, every rotation has a purpose, all working together to generate force and movement reliably and robustly. It really deepens your appreciation for the complexity humming away inside ourselves.
It really does. Which makes you wonder, what other biological processes, things we only understand from diagrams right now, are just waiting for the right technology to reveal their dynamic beauty?
That's the big question, isn't it? And how might understanding these molecular tricks, like that chameleon grip of myosin securing itself before moving, how could that inspire us? Maybe in designing nanoscale machines or even finding completely new ways to approach drug development.
So much still to explore in the tiny world that makes us work. Fascinating stuff.